First, many thanks to Jessica and Amber for initiating and organizing this symposium. The question I address is whether early life factors affect us through creating stable individual differences in the brain, or if they work to make our brain change differently through the lifespan. I have no conflicts of interest to declare. I come from lifespan cognitive neuroscience, and in the field of brain aging, there is much focus on how people may decline differently and how we can affect slopes, that is, trajectories of change. But what struck me early on in working with brain images and age changes was not the individual differences in change, but the persistent individual differences seen over time, even in the presence of gross age changes. Brains differ a lot and they all change, but I think it's not so easy to tell if they actually change differently. As an example here, you see ordered along the y-axis the brain scans of 5 individuals in their 70s followed in my lab for a decade, with 3 time points along the x-axis. It is easy to see that they differ to begin with and that they change in aging, for instance with widening of the ventricles. But it's not obvious that they change differently, and the variance in offset far exceeds the variance in change within this time frame. So they look like themselves all along, and not like others at the same age. I will revisit this similarity to oneself across time later, but for now, let's just briefly see what this offset or level versus slope or change means in terms of conceptual lifespan differences. So differences in brain and cognition at any point in life reflect variation in previous level, how the brain has developed to begin with, and differences in change. Differences in level or offset are captured in the concept of brain reserve as shown here in panel A, and differences in change in concepts such as brain maintenance in aging shown in panel B, or mental age and catch-up in development as shown in panel C. Differences in level and change may also coexist and interact, consistent with, for instance, the Matthew principle. All of these different scenarios can result in people falling below a functional threshold at different ages, but they have very different implications regarding prevention and intervention. I argue that while individual differences in level of brain and cognition in normal populations are substantial, there is little evidence to conclude that these are caused by differences in change. Rather, there is much evidence for stability of early life effects through the lifespan. Even though brains change much with age, we and others have documented stable associations of early life factors and brain structural characteristics. In terms of genetics, one example I believe where quite a few have taken a brain maintenance account as a premise is the case of ApoE4. Here, an antagonistic pleiotropy account has often been drawn upon, assuming that having the risk allele has no or even possibly some positive effect in young and reproductive age, but the risk is supposed to hit people in older age, and someone will make their hippocampi atrophy faster. However, we investigated this in a lifespan sample of nearly 1,200 persons with more than 2,600 brain scans and found that higher polygenic risk score for Alzheimer's disease and presence of ApoE4 were associated with lower hippocampal volumes to begin with as offset effects rather than faster decline in older age. So having higher genetic risk was, as predicted, associated with lower hippocampal volume in 80-year-olds, but we saw similar effects in development and young adulthood. Not only genetics are associated with brain characteristics in a stable manner, we may see similar associations for early life environmental factors. So we did the first quantitative MRI study of children exposed to opioids and polydrug use in utero and found lower neuroanatomical volumes virtually throughout the brain, in part linked to differences in cognitive and behavioral scores. These differences in the opioid-exposed children were persistent when we followed them up again as young adults nearly a decade later. And such differences among the groups likely don't just reflect genetics, as when we have studied children born to mothers addicted to opioids but detoxified in a hospital setting, we did not see such differences in brain and cognition. And whereas the children exposed throughout pregnancy also had significantly lower birth weights than controls, the birth weight of the children born after detoxification was not significantly lower than that of controls. Average lower birth weight has been reported in a range of neurodevelopmental disorders, and birth weight itself is an interesting metric. We have documented that associations of birth weight and cortical area and volume are found at all ages with very high spatial correlations across our lifespan sample, the aging UK Biobank, and the developmental ABCD sample. However, no consistent spatial correlations are found for effects of birth weight on slope or change in these longitudinal datasets, testifying to these being stable effects on level of cortical volume found throughout life, regardless of age. One may, of course, also ask what portion of this reflects genetic influences, but birth weight discordance in genetically identical twins yields similar effects as seen here. So it appears likely that environmental influences in the womb work to affect lifespan level of brain features. I will argue that in terms of environmental influences, effects on the brain in utero while being formed often far exceed effects of environmental factors in adulthood. I was invited to write a review for Trends in Cognitive Sciences on the dynamics of brain health and cognition through the lifespan, and I and my colleagues Martin Løvden and Anders Fjell chose to focus on the timing of lifespan influences on brain and cognition. In this forthcoming paper, we compare effect sizes of modifiable environmental influences on gross brain volumes for different life stages. The background for this is that in 2017, the Lancet Commission for Dementia Prevention, Intervention and Care published a life course model for effects of modifiable factors. And this was updated in 2020 to now include 12 modifiable risk factors, the modification of which they claimed can delay or prevent 40% of dementia cases. The two Lancet papers have been cited more than 5,000 and 4,000 times respectively and have received a lot of attention. And they are to be commended for stressing a life course model, but the reality is that most of the targeted factors are in adulthood, the only exception being education, belonging to late childhood and adolescent stages. Our message is that this is unfortunate because, as you can see from our calculations, effects of potentially modifiable factors on the brain appear much greater at very early life stages. Please note that the scales for these two panels for fetal and later life risk factors are different, and the average effect size on the brain of the modifiable risk factors called out by the Lancet Commission for Dementia Prevention is indicated by the stapled line here. So the effects of fetal life risk factors such as opioid, alcohol, and tobacco exposure, as well as prematurity and very low birth weight, are many times as big as for any such factor in adulthood or aging. In view of this and the apparently stable offset effects on level of brain characteristics through the lifespan, I find it pivotal that we try to understand also how early life factors work on brain characteristics, change, and plasticity in adulthood and aging. And this is what we seek to do in my ERC Consolidator project, Set to Change, which unfortunately has been hard hit by corona restrictions, so it has not been powered or finished as planned, but I would like to show some preliminary data. The aim of this project is to identify how early life environmental and genetic factors restrict and promote neurocognitive plasticity through the lifespan. So we experimentally test differences in brain and cognitive plasticity by a crossover design with resident training of adult mono and dizygotic twins of varying ages and with varying prenatal environmental differences as indexed in birth weight, on average by the discordance is about 9% between monozygotic twins, which is close to the about 13% average difference among dizygotic twins. So we get information on birth weight from the Norwegian Twin Registry and the Norwegian Medical Birth Registry. To max training effects, we have a 10-week navigation training paradigm in an immersive virtual reality, which I'll show you. But first, the fantastic team, which has been doing and is doing an amazing job. So the twins are immersed in a town we have made called Plasticity, and they have to learn to navigate an intricate area of about 6 square kilometers. And first they are finding targets, and then they are also getting these retrospective and prospective memory and time tracking tasks. New areas of the city are sequentially unlocked with improved performance. For instance, if they get to the end of the world by the sea, they will be rewarded by coming to Kristine's Bait and Beer store, which is a place that has long existed in my mind and I wish would exist IRL. So here you also see an example of the retrospective memory tasks. What were you to get at this palace? And we have created alternate worlds to test their navigation performance before and after the intervention and before and after rest. So here you can see some more of what the setup looks like. If you wanted, let's see. Yeah, so I wanted to have real life locomotion, but VR treadmills are very infeasible in our lab settings. So we have them cycle on bike rollers while we are wearing VR headsets. And we have trackers on the bikes, of course. So when she gets to this target, she also gets feedback on how much she deviated from the optimal path. So if we go on, here is another movie of how it looks from outside, giving an idea of what kind of rich data we can get, or just about how they move and what they look at and so forth. And this is just a bird's view. So now I'll move on to see if our participants' brains deviate before and after training by early life factors, including genetic similarity and environment in the womb, as indexed by birth weight, we compare their anatomical fingerprints from multimodal imaging, in this example, T1 and T2 weighted scans, to create our measure of brain similarity across the individuals and the time points. Then we process these scans using the free surfer longitudinal stream, and we use cortical thickness, volume, and area of 68 regions, as well as the SAMSEG subcortical volumes and coarse morphological measures, excluding tissue types other than gray matter, white matter, and cerebrospinal fluid. So the resulting brain print is a vector of 239 features, each normalized relative to sample mean and standard deviation. And pairwise similarity between brain prints is established via cosine similarity, providing a robust indication of the similarity in the pattern of anatomical structures between individuals and time points. So in the movie to your left, you see alternately the brains of two co-registered monozygotic twin pairs, one younger pair on the left and one older pair on the right. And as you can see in the movies, global brain differences, even between monozygotic twins, are evident, but our measures still capture subtle similarities. So the plot to your right shows similarity over 2 visits, separated by 10 weeks for the sample so far analyzed, including 43 monozygotic and 27 dizygotic twin pairs. And as you can see in the main diagonal, self-similarity is very high and can be used for perfect identification. So the two other clearly visible diagonals represent monozygotic similarity, that is between subject similarity between twins. The two faint lines can also be seen here representing dizygotic similarity. So how does birth weight difference between monozygotic twins affect their brain print similarity as adults? These twins are now in the age range 16 to 78 years, and mean age is about the mid-30s here. As seen from the plot, at baseline MRI, before training, the greater the birth weight discordance among the monozygotic twins, the less similar are their structural anatomical brain prints, whereas no similar trend is seen for the dizygotic twins. So we have significant improvements in navigation after training relative to after rest, but with much variance. And an important question is if birth weight discordance also affects brain plasticity. We calculated functional connectivity across the whole brain pre and post training to see how much an individual's functional connectome changes with training. So more change can be an indicator of greater plasticity, we think. We calculated the within pair differences in connectome change by subtracting the low birth weight twins change score from the high birth weight twins change score. And so far, we find that the extent of change varies with birth weight discordance within pair, where the heavier birth weight twin shows higher connectivity change with training. So given these brain structural and functional differences linked to birth weight differences, in monozygotic twins, it is natural to ask the question, to what extent intervention in adulthood can still influence trajectories? Can the low birth weight twin catch up with the high birth weight twin following training? As in this project, both twins in a pair train at the same time, we cannot test this simply by comparing post-training similarity with baseline similarity. So in this analysis, we let the heavier twin remain at baseline and test whether the lighter twin gets a more similar brain print to the heavier sibling after training. We test similarity change as a function of training time put down by the low birth weight twin. So here in 24 monozygotic pairs, we find a significant positive effect of training time on increased similarity of structural brain prints with a correlation of 0.42 in monozygotic pairs. So for the next year, we're going to try to complete data collection, also with follow-up across years. And so far, my take-home messages from the observational studies by us and others, as well as from the experimental twin study, is that first, we must never ascribe differences in adulthood and aging to differences in change with age without knowledge of what came before or what differences were there to begin with. And second, we see consistent and stable effects of early life factors. Differences in level or offset early on are seen through life, while we so far have less evidence for differences in trajectories of brain change. I think an important challenge now is to further test how early life factors affect the brain and its plasticity in adulthood and aging. So far, the data tell us that we need to attend to timing of influences on brain and cognition, and such influences work from the beginning of life, for life, and all through life. And by this, I just want to thank all my lab, all my excellent collaborators, and all of those who work to get data collected around the world. Thank you.